This module will cover the neuronal addition of cells, their migration processes, and the aggregation to other cells. As we learned earlier, by the 24th day after conception, the neural tube has already been formed. Once the neural tube has formed, then neural proliferation begins. Proliferation is nothing more than the great increase in number of cells. We find that the cells of the tube would begin to increase in a fashion that is quite impressive. A study done in 2008 by Lumen revealed that the embryonic development of a nervous system is occurring at a rate of 250,000 neurons per minute. This would allow the species to develop enough neurons, which eventually some of them are going to die, and we'll talk about this in a future module. But please note that the cells proliferate in a way that is consistent with a specific species. In the example of humans, we will see 3 swellings at the anterior end, and these will eventually become the forebrain, the midbrain, and the hindbrain. Please note that the proliferation process is quite complex, and it is in part controlled by chemical signals that come from 2 organizer areas in the neural tube, and this is what we call the mesoderm. We call these the floor plate, which runs along the midline of the ventral surface of the tube, and then the roof plate, which runs along the midline of the dorsal surface of the tube. Once enough cells have been created through cell division, they begin to migrate to an appropriate target location. During the period of migration, the cells are still in immature form. They lack the processes such as axons and dendrites that characterize mature neurons. 2 major factors govern migration in the developing neural tube. That is time and location. And that is important for us to note. In a given region of the tube, subtypes of neurons arise on a precise and predictable schedule, and then they migrate together to their prescribed destination. And that is why we look at, again, the 2 major factors that govern migration being time and location. Some individuals believe that there are greater principles that are operating behind the scenes that are responsible for where you are at any given moment. And that's why some individuals say I was at the right time at the right place. Some people say I was at the wrong place at the wrong time. And if you'd like to compare this to the migration, we find that if the cells would arrive too soon and they migrate too early, they just may not be able to survive, as there wouldn't be any other cells that they could form bonds with. Another thing is that if they migrate, but they migrate the wrong place, they would also not be able to survive, bringing about complications to the organism. In this picture, we're able to appreciate the neural tube here in green, and you're able to see 2 types of neural tube migration. You have radial migration, which would be the blue ones here, and these are basically those that are moving out from the ventricular, each side of the 4 connected fluid-filled cavities in the center of the brain. This will eventually become our ventricles. And they're moving in a straight line outward toward the outer wall of the tube. The other type of neural tube migration is tangential migration, and that is where they're moving up. And by tangential, this would occur at a right angle to radial migration, that is parallel to the tube's wall. So you'd be looking at it in a pattern that's like this, consistent at the bottom and on the outer purpose. There are also 2 methods of migration, and we look at somal translocation and glial-mediated migration. With somal translocation, we find that an extension grows from the developing cell in the direction of the migration. The extension seems to explore the immediate environment for attractive and repulsive cues as it grows, and then the cell body itself moves into and along the extended processes. This process brings about like a trailing fashion and eventually is retracted. And it's almost kind of like a snake covering some ground and making sure that it's going through the right place. The glial-mediated migration is one that moves along the radial glial network. And that would be that once a period of neural proliferation is underway and the walls of the neural tube are thickening, there is a temporary network of glial cells. And these radial glial cells would appear in the developing neural tube. At this point, most cells engage in radial migration to do so by moving along this network of cells. Please note that most cells engage in both types of migration, and many radioglial cells eventually develop into neurons. Here we can appreciate the somal translocation, and that could be in either a radial or tangential form of migration within the tube. Here you can see how the cell started off like this, and then through its projection is able to grow an extension. And then once it detects and is going around through chemicals being guided, it covers quite a distance. Eventually the body begins to catch up to it, and just like that it would have migrated to another form and to another location. The glial-mediated migration, this would only travel through radial networks, and these right here would be the radial glial cells. The neurons would bind to these glial cells, and through this process they would begin to migrate to the specific location where they'll eventually end up. Please note, as stated earlier, that these glial cells that we have here eventually develop into neurons. And so we find that these would also eventually go through a transformation process. They morph into a neuron. Please note that the migration is guided by various chemicals. They either attract them or repel them, and based on this in here, the guidance of molecules would play a critical role on the development of our nervous system, because the brain cannot function normally unless each class of developing neurons arrives at the correct location. In this specific slide, we'll be looking at the neural crest. Please keep in mind that we are always able to appreciate the neural crest at 18 days after conception. You can see it here in its purple form. And then eventually it starts forming at the dorsal part of the neural tube. The neural crest cells develop into the cells of the peripheral nervous system. So this is what you and I will start looking at aside from the spine and brain, the spinal cord and brain. This is what makes up the rest of us. All the nerves, all the afferent nerves that allow you to respond to stimuli on the outside actually come from the neural crest in this piece right here. The cells do migrate long distances, as we do have quite a bit of space to cover. The migrating neurons, again, are guided by these chemicals that either attract or repel them. If they're a specific type of cell which has developed into, as stated earlier, a specialized cell, it would then be guided by chemicals and attracted to a specific location. Anything other than that, or any type of miscommunication with chemicals, would end up with would result in a very odd looking body. Think of your body and think of the skin in your lips, perhaps, in comparison to the skin in your eyelids. Had the cells that were supposed to be designed for your lips migrated elsewhere, you would end up with a really different looking structure. Looking specifically at the brain and looking specifically at these cells, we know that the cells of the hippocampus and of the amygdala and the cerebellum and the pons, although they're all similar in various ways, in order for them to function well, they need to arrive at this specific location and at the right time. This would bring about what we would call normal human behavior. Any variation from this in here can result in difficulties with our behavior and mental processes. Once the developing neurons have reached their location, they have to align themselves with other developing neurons that also migrated to the same place. This would eventually give way to forming the structures of the nervous system, and this is what we call aggregation. The elimination of just one of these molecules that would allow for the aggregation can actually bring about a devastating effect on brain development. CAMs, which are cell adhesion molecules, are located on the surfaces of neurons and other cells. These aid both in migration and aggregation, and they also recognize and adhere to molecules on other cells. This should be similar to the concept of a sticky note. Please note that adhesion actually does come from that of adhesive process, a tape. The gap junctions are points of communication between adjacent cells, neurons and glia. The gaps are bridged by narrow tubes called connexins, through which the cells can exchange cytoplasm, and that is the material that lives inside the cell with the exception of the nucleus. The cytoplasm is where most cellular activities, like the division, occurs. It also contains a lot of the organelles and bacteria. So these gap junctions in here would are prevalent in the development of the brain, and they play a very important role in the migration and aggregation and other processes.